Hi, I'm Haley McMillan, and this is a video on the cell cycle. The cell cycle is highly regulated and has 5 stages. The 5 stages of the cell cycle are G1, shown in blue, S phase, shown in red, G2, shown in green, M phase, shown in purple, and G0, shown to the right of the diagram. In G1, the cell is growing and developing. In S phase, synthesis and replication of DNA is occurring. In G2, the cell is growing and preparing for mitosis. In the M phase, PMAT, also known as prophase, metaphase, anaphase, and telophase will occur, and after that, cytokinesis will occur where the cell will officially split into 2 identical daughter cells. Now, G0 is kind of weird. G0 is what happens when the cell basically messes up in G1. After the cell messes up in G1, it cannot move on to S phase, so it has to go to G0 phase where it will not be able to divide. The cell cycle has checkpoints that regulate, the cycle. Okay. 3 of the major checkpoints are in G1, G2, and M phase. A checkpoint is a control point where stop and go signals can regulate the cycle. Now, as you can see, the little men on this picture aren't exactly accurate, but my arrows are. And you should trust the arrows, and you should trust me because, we've established that kind of trust. Now, molecular regulation. Molecules regulate the cell cycle. The molecules are generally protein kinases and cyclins. Protein kinases activate or deactivate other proteins by phosphorylating or removing a phosphorus. Phosphorylating them, and it can even give go-ahead signals in the G1 and G2 checkpoints. Many of the kinases involved in the cell cycle must be activated by cyclins. These are called cyclin-dependent kinases, or CDKs. As shown in the diagram to the right, the red bean-shaped pictures are the CDKs. I'm going to use the blue one. The blue S-cyclin is obviously the cyclin. The red CDK will not activate until the S-cyclin reaches it and attaches. Only when that attaches can the cell cycle continue on. MPF. The maturation promoting factor, or MPF, is a CDK complex that signals a cell's passage from the G2 checkpoint to the M phase. Now, these are sometimes called the mitosis promoting factors because of what I just said. As shown right here in the diagram, the MPF helps the cell move from the G2 checkpoint to M phase. PDGF. The PDGF, or platelet-derived growth factor, is made with blood platelets. When injured, or when you're injured, platelets release the PDGF, and the PDGF helps to heal the wound. If you were wondering, a growth factor is a protein released by certain cells that stimulates other cells to divide. This is a picture of a PDGF, obviously. Now, cancer cells. Cancer cells are really bad, and if you don't know that, I'm gonna make it a little more relatable for you. Cancer cells are the kids at school who pick on others, don't do their homework, don't listen to their mom, little stuff like that. Those are cancer cells. Please do not be a cancer cell. They're just, too cool for school, and they break the rules of the cell cycle. Not cool. Next, this is a picture of basically what a cancer cell is. Cancer cells occur when basically a cell goes off the edge. Okay, a cell goes off the edge and does whatever he wants. It goes crazy, right? It says, you know what? I'm gonna divide right here, and I'm gonna make more of me, and we're just gonna dominate and ruin people's lives. So here it is, right there. Okay. Now there are a few things that separate cancer cells from normal cells. For one, cancer cells do not need growth factors to grow and divide. So, they do what they want, or they make their own growth factors because they're just macho like that. And two, cancer cells stop dividing randomly and can divide indefinitely with the right amount of nutrients. This picture to the right is a picture of just a really gross looking cancerous cell, or cancerous cells dividing. Kind of looks like snot balls. I'm sorry. Oops. Okay, let's go back. When cancerous cells are not destroyed, they may form tumors. A tumor is a mass of abnormal cells within otherwise normal tissue. Benign tumors may grow, but they don't spread or cause any serious problems. They can be removed surgically removed. As shown on the left side of this picture, benign tumors are withheld inside a capsule in which the normal cells are contained, and they can't really go anywhere. They're just stuck. It's like cancer gel. Cancer gel. Yep. Malignant tumors, on the other hand, can spread through a process called metastasis, where, like I said, they spread and can impair the functions of other organs. These are the more harmful ones that can really only be solved, fixed, or gotten rid of through chemotherapy, which targets basically the DNA of cancer cells. Now, I'll let you decide, but for me, if I were to ever, develop cancerous cells, which let's hope I don't, I'd probably want a benign tumor because it is the lesser of the two evils. Now, that was the cell cycle. I'm Haley, and I hope this was helpful.